As we all know, our DNA is the genetic material that makes you, you. According to the central dogma of biology, DNA is composed of the 4 nucleotides, A, T, C, and G, that are read and transcribed into RNA, which is then translated into proteins that affect how our bodies function and how we look. The DNA sequences that encode these specific physical characteristics are called genes. However, if these sequences are disrupted by various types of mutations, the normal gene function can change and cause certain physical changes, including diseases. For example, cystic fibrosis is a rare disease with no cure that results in the fluid buildup in the lungs over time. There are over 1,200 known mutations in the gene that causes cystic fibrosis. However, cystic fibrosis is only one of the many diseases that is caused by a mistake in the genetic code. Therefore, developing a way to somehow fix these mistakes to help cure or treat disease is one of the biggest problems that scientists all over the world are studying. For more than 40 years now, scientists have been inventing genetic engineering technologies to find new ways to try and treat diseases at their roots, their DNA.
Just do it! Don't let your dreams be dreams.
The first recombinant DNA molecule was created in 1972. Recombinant DNA involves bringing together DNA from multiple organisms and creating sequences that would normally not be found in their genome. A year later, researchers created the first organism with recombinant DNA by successfully introducing recombinant DNA into bacteria. The first transgenic animal was generated in 1974, where viral DNA was introduced into the mouse embryo. There were flaws with this method though, since DNA integrates randomly into the genome and not at a specific site. In 1990, the first transgenic human was created. Sometimes this technology worked, but sometimes it could be dangerous to the health of humans. Later, homologous recombination was also used to try and edit genes. It involves the exchange of nucleotide sequences between 2 similar molecules of DNA, essentially switching a good gene and a bad gene. Overall, homologous recombination had drawbacks including low efficiency, high costs, very time-consuming and technical, and did not work in all organisms like humans. Other gene therapies included zinc fingers and talons. These technologies utilize sequence-specific DNA binding proteins. Talons and zinc fingers require expensive engineering of these proteins to achieve sequence-specific binding. These methods also have generally low accuracy and are time-consuming. This increases the need for a genetic modification method that is easier to use and is overall more efficient.
Tell me how!
However, while we were looking elsewhere for the answer to gene editing, the solution was there all along, hidden inside bacteria that have existed for millions of years.
What? You gotta be kidding me!
In 1987, scientists discovered an unusual stretch of DNA in E. coli bacteria that consisted of identical repeated sequences with spacer DNA sequences between them. These repeated sequences were present in bacteria everywhere, so scientists concluded that they must have an important role in bacterial functioning. However, it wasn't until 2005 until the technology and data were available to find out just what these sequences were. To their surprise, the DNA wasn't bacterial DNA at all. These bits of DNA matched to viral DNA. How could that be? It turns out that bacteria have a defense system where if they survive an attack by a virus, they are able to take a piece of the broken viral DNA and put it into their genome so that if they are attacked by the same virus again, that DNA is transcribed into RNA, which is then able to fight off and kill the incoming virus. Essentially, this awesome system acts as a DNA detective, searching for any suspicious intruders with the help of their DNA mugshots that they keep on file from last time. Scientists have called this defense system Clustered Regularly Interspersed Short Palindromic Repeats, or CRISPR for short. So, how does this work? When a bacteria is attacked by a virus, a gene for the Cas9 protein is transcribed and translated, coinciding with the transcription of the matching mugshot of viral DNA stored from last time, called the guide RNA. The Cas9 protein is a type of protein called an endonuclease, which are able to cut DNA sequences at specific locations. The Cas9 protein then associates with the RNA mugshot by binding to the tracer RNA that is associated with the Cas9 protein. The tracer RNA is a complementary RNA molecule to the guide RNA, allowing them to bind with each other. The guide RNA is a perfect match for the viral DNA and goes searching through all the other molecules until it finds its matching suspect.
I think we won. What were we trying to do?
When it finds the culprit, the guide RNA binds to the viral DNA, activating the Cas9 protein. Cas9 acts as a kind of molecular scissors as it cuts the DNA on both strands, causing a double-stranded break, which effectively kills the virus and allows the bacteria to survive. However, in 2012, Dr. Jennifer Doudna at the University of California, Berkeley had an idea. This natural defense system could be refashioned into an incredible tool for genetic engineering. We can make the CRISPR-Cas9 system cut whatever we wanted to by simply replacing the guide RNA with our own DNA mugshots. After it cuts out the gene we don't want, all we need to do is put a new gene in the general vicinity, and the homology-directed repair mechanism in cells will incorporate it into the DNA. Homology-directed repair is the preferred method of DNA repair compared to its counterpart, non-homologous end joining. Homology-directed repair allows for a more accurate repair without the risk of adding in or getting rid of the wrong DNA bases. Today, CRISPR is being widely used in research as a way to cheaply and efficiently edit DNA instead of using previous methods that are less effective and more expensive. Currently, CRISPR is being used to quickly and efficiently create animal models for researchers in order to help figure out the mechanisms of certain diseases that are still unknown to us. In addition, while CRISPR can be used to fix mutations or introduce new DNA sequences, it can also be used to cause mutations in the DNA or knock out a gene entirely. In doing so, we can further understand how changes in the genome affect the phenotype of a gene. Also, we can better understand protein structure, their mechanisms of action, and their functions if still unknown to us using CRISPR. Inactive CRISPR can also be used for DNA labeling and regulation of transcription, as its binding mechanism is specific to only certain sections of the DNA. As a result, there is no more powerful gene editing tool than CRISPR. It is simple, fast, cheap, and highly specific. Its utility is unmatched, and it has been shown so far to be a universal tool for gene editing in any organism or species, making it a truly revolutionary technology that can alter the course of human history.
That's one small step for man.
Individuals have been genetically modifying crops for thousands of years through crossbreeding and artificial selection. In artificial selection, scientists continuously breed plants for desired traits. For example, corn originally started as a long, lanky plant, but through selective breeding it became shorter and more round. Eventually, as you have learned from previous videos, companies such as Monsanto began selling seeds that were genetically modified to be pest or herbicide resistant. For example, scientists were able to incorporate a new gene from a bacterium that produces toxins that kill pests when they try to eat the plants. CRISPR can serve as a faster, more precise, cheaper, and easier way to add or remove genes in crops than traditional breeding methods or other technologies. CRISPR can make plants that are higher yielding, disease resistant, more nutritious, or simply just better tasting. For example, a biologist at Penn State University is using CRISPR to create mushrooms that do not turn brown at the slightest touch, and did so by knocking out a browning gene using CRISPR.
I'm not even mad. That's amazing.
Inserting DNA or disrupting a gene has become a relatively simple task, as all that researchers need is a Cas9 protein and an RNA guide. One challenge that faces CRISPR's ability to modify crops is that plant genomes are highly repetitive, which increases the likelihood of off-target cutting.
In the future, we hope to use CRISPR to fix disease genes in adult humans. CRISPR would be used to edit out the defective version of a gene and replace it with the healthy version of the gene.
No way, Jose!
By replacing the mugshot with a complementary copy of the mutated portion of the gene would be targeted and cut out. The new, healthy gene must simply be put in the same vicinity, and through natural repair mechanisms, it would be put in the newly vacant site using homology-directed repair. These modifications could cure the disease or reduce the risk of developing a disease. Genetic diseases that are most amenable are ones that only require a single allele to be targeted. Common diseases that could be treated or cured using CRISPR are muscular dystrophy, cataracts, cystic fibrosis, HIV, Huntington's disease, and malaria. Editing adults versus editing embryos is very different. Adult humans are composed of trillions of cells, whereas embryos are only composed of a handful of cells. This means CRISPR will have to edit the genes of many more cells in an adult versus an embryo. There is also the possibility of creating a mosaic where only some of the cells get edited. In adults, only the body cells are altered, meaning that the altered gene would not be passed down to the progeny. Changing an embryo's genetic material would cause the modified trait to be passed down for generations and possibly interrupt genetic diversity, but would potentially rid the family line of the genetic disease. When editing an embryo, there is also the ethical issue of not having consent because the change could potentially affect the embryo's future offspring. Currently, no embryos that have been modified using CRISPR have been implanted in a mother, causing an ethical dilemma for using embryos in the research because after the study, they are simply discarded. This would conflict with many mainstream religious ideals, calling into question the use of CRISPR in studies and various treatment options.
While CRISPR is a new and growing technology, scientists worldwide are already researching and using it. Recently, CRISPR was used in an experiment performed on mice with muscular dystrophy. Muscular dystrophy is a disease that causes progressive weakness and loss of muscle mass. These scientists used CRISPR to fix the faulty muscular dystrophy gene. Even though the mice did not become normal, they saw the mouse's general strength grow and heart become stronger over time. Another incredible procedure was recently performed on humans by doctors in China where they used CRISPR to hopefully treat an aggressive lung cancer in an adult male. An aspiring future application is a CRISPR pill, which is a pill that has the potential to replace modern antibiotics. Instead of taking antibiotics when you have a bacterial infection, you would take a pill full of CRISPR that would not only eradicate the infection, but also make sure you never get that infection again.
Yes! I am invincible!
A prominent problem in our culture is the lack of organs for organ donation. However, pigs are easily bred and are available so that using CRISPR, a pig liver can be altered to create a human-like liver that will be less likely to be rejected in a human body. This would generate even larger organ banks to help more people who need organ donation. Another cool use of CRISPR for the future would possibly be resurrecting long-lost creatures. Scientists have the potential to take woolly mammoth DNA and insert it into a living relative such as an elephant and create a living woolly mammoth. Another potential CRISPR technology would be editing the genome of a chicken and creating a creature that looks like the ancestor of dinosaurs.
That's not made up, that sounds made up.
While CRISPR technology presents many benefits for the future, such as eradicating diseases and allowing for a generally healthier population, there are also many concerns. Some social concerns are that only wealthy people would be able to afford it, creating a large divide between the rich or the healthy and the poor or the unhealthy. Also, CRISPR has the potential to be used as a weapon, where if fallen into the wrong hands, it could be used in toxic ways to spread disease. CRISPR could also possibly lead to the creation of a superspecies which could be resistant to any disease or illness.
I think I just peed a little.
While these fears are extreme and most likely rare, the power of CRISPR still presents great concern. The underlying theme of these concerns is that CRISPR is irreversible, and if you use it to change one person, all of their future progeny will be affected. This brings up the issue of consent and how these affected future generations are not able to give consent for using CRISPR to alter their genome. People could also use CRISPR technology to create designer babies. This would mean that prospective parents would go to the doctor and request certain traits and appearances that they want their children to have. This would lead to a disruption of genetic diversity and more potential dangers. Overall, the growing and fascinating field of CRISPR technology still has a long and challenging path ahead, but it is starting to pave the way for an evolving future. Compared to previous technology, CRISPR can revolutionize how scientists edit DNA. In the future, CRISPR could be made to modify agricultural plants, fix disease-causing genes, and eradicate diseases. Although there are numerous awesome applications of CRISPR, it is important to understand that the technology is not perfect and still being developed. While the CRISPR technology is outstanding and revolutionary, it comes along with numerous ethical concerns. These concerns will guide our society to approach the use of CRISPR with great caution and care. In the near future, we hope to be able to use CRISPR in a safe, controlled way to prevent and heal diseases in order to create a healthier population.